So very good afternoon to everyone. I'm very much pleased to see all of you, and also welcome to all colleagues from our other locations. It seems that right now we have only one location active. 3 locations? Yeah, that's good. So welcome. And I think that this year we have not been very proactive for the CFA meetings because of the range of the regions that, although means whenever this was possible we organized, but somehow things did not, they were not that active as we were there last year. So last week I told Anu that she needs to organize the meeting as early as possible. She says we need some speaker at the same time in our lab. Many times, especially in our lab and also some other colleagues, they have always say that, well, it will be good idea if you can suggest some idea to write the paper. So this doesn't mean that if I'm giving the talk that I'm the best person to give you the advice, but what we can do, we thought that, yeah, this will be good idea because we have many new colleagues, and sometimes even when I'm working with you, I'm always having this difficulty, so that, well, how to move ahead. So we thought, let me share our experience, what we are doing, and of course different colleagues, they have the different kind of experience, so we can have some good discussion on this topic. So that's the reason that I volunteered, or I was forced to volunteer to give this presentation. So that's the reason we are here. Anyway, before we start, let me say a few welcome words to some new colleagues who have joined. For them, this is the meeting for the first time. So tell me that, can you raise your hands that for, okay. Yeah, so you can introduce yourself. You can keep on sitting, so we are very informal. Yeah, so please, yeah.
Vishwajit Mandal, IAPR country.
And you are here for?
I'm here for 3 months attachment training.
Attachment training, okay. So you want to be attached, this is good.
I'm attached to the chickpea breeding unit.
Very good. Okay, next. Sandeep, you raised your hand? Yeah. Okay. You can also grab this mic from here, yeah. Now we got more people, it seems.
Myself Sandeep Kale. I joined in March as a special bioscientist on chickpea genomics.
Okay, good. Fida.
I'm a research associate at Gene Bank at ICARDA. I joined here just 1 week ago. I want to do my PhD with Dr. Rajeev, mainly on chickpea.
Okay.
We will see if we can just
try
to propose it today.
Okay.
And you have the ideas today.
Okay, good. Thank you very much. So welcome to all the new colleagues who have joined this group. And I know my request is that you can add their names in your list. And of course, we have Dr. Noel Ellis, CRP Director on Grain Legumes, as a resource person in this meeting. And I think that all other people, they know each other very well, right? Anybody from our other locations, we got some new colleagues there, or everyone is, or miss, all old folks are there? Ankit. No new person? Okay, good. Ankit, yeah, so Ankit, stand up here, you introduce, please come here.
I'm Ankit Jain. I'm Ankit Jain, and I'm in project administration, so I joined a month back.
Good, welcome, yeah. Thank you. So what we will do, that we will have this presentation and we can have the discussion in between, or maybe that let me go through that, we can have the discussion, we can learn from you, so you can give some idea. And none of us is perfect, this is my feeling. We always can learn from each other. So this is the thing. And after that I will also give, Manish is there now, Rokiwal, or not yet? Okay, yeah. So after that I will have another 5-10 minutes presentation, and we hope that we should be able to complete in 1 hour. So maybe I can go there. So the first thing is that, well, of course, and let me present again that this does not mean that when I'm giving the presentation that I want to emphasize that, well, people should work only for the papers. Our context, all of us know that we are working at ICRISAT, and what is our mission? Our mission is to produce the international public good, or we can help the poor farmers in developing countries in sub-Saharan Africa and Asia. But the point is that we are science-based organization. So when we are doing scientific research, we are producing a lot of stuff. Now the question is whether we need to share it with the public or not. And I think that scientific publication is one of the several means to share your results with other colleagues. This does not mean that scientific publication is everything, or I am giving lot of emphasis on scientific publication. So this is, I am already putting this disclaimer. but we know that well what is our goal. This presentation is just in context that of in the context of the in the interest of the young scientists or colleagues who would like to publish or who are starting their career. Otherwise, many of us already published good papers, so this is good. So, and we can learn from each other. So, what first thing is why we need to publish. I think that nowadays many funding agencies, and when you are having when we are science-based organization, then we are getting the funds from the international organization. Then this is an obligation to those funding agencies that what we are doing, we are producing results or so, we need to share. So this is, in my opinion, this is obligation. When you are a scientist, so again, don't say that well my obligation is just to publish the papers, not to produce the varieties of pre-breeding lines. So I want to have this thing again and again very clear. And all of us are having the different kinds of roles and responsibilities. For some of you, this may not be applicable at all. For some of you, this may be applicable. So have this thing very clear in your mind. Sometimes we say, and I am giving the example that people say whether we should publish the positive results or negative results. In my opinion, if you were failed in some experiments or if you did not get the exciting results, then in my feeling also you should publish that one. For instance, we know that in the case of peanut, molecular marker polymorphism is very low. Now, for instance, if you screened 2000 SSR markers, you found only 50 SSR markers polymorphic. On the other hand, somebody who was working on rice, out of 2000 they got 1000 polymorphic, and you started to be disappointed. My God, I got only 50 markers polymorphic. My feeling is this result is also important result for the community, so that they know that well which 50 markers they are more exciting, or they are having higher probability if you screen the large number of germ plasms, or the markers which did not show any polymorphism or there was some problem of the amplification, etc. So what I am trying to say is that even those results are not very exciting, but they will help community that they should not be using or they should not be using that approach. This does not mean I am asking you that we should be publishing the rubbish results. So we should have very clear difference, negative results and rubbish results. If you have done something wrong with your experiments and then also you are getting the failure, then you should not publish that one. So I am saying that we should be okay with positive and negative results, and nowadays there are several journals, they are also interested to publish your negative results. So this is important. Third thing is when you are working in an organization like ICRISAT, when you are publishing, this also brings the visibility to that organization at international level. Many times you keep on doing the work, when someone talks to you, they say, wow, you are doing great work. But then you could communicate this work only with the 2-3 people, 10 people with whom you were talking. When you put in the public domain, you are sharing this thing with everyone, and then those people also understand, wow, ICRISAT is doing this work. So this is also important for bringing international visibility to any organization. We are working for the organization. So our first priority is to fulfill the mission and vision of the organization. At second point, then you come as an individual, but first thing is for organization, that what we are doing for the organization. But when you are publishing this work in parallel, in addition to the working for organization, you are also enhancing your reputation. You are also enhancing your visibility at international level, that wow, Rakesh Srivastava is great scientist, he published 10 papers in pulp millet, he is doing thing, Janila is doing groundnuts, so like those kind of things. So again, you as individual, in my opinion, come at the secondary level. First level is the institute. So now, and when you are working, so what I am saying that this enhances scientific reputation of the scientist. Nowadays there are different kind of metrics. I am not going to discuss. There are some people they talk for the citation index, H index, impact factor, etc. There is another group of people, they say this is rubbish. So I am not going to discuss those things. Anyway, in international community, there are different kind of, they call it scientometry or something like that. So yeah, so then you are having the different kind of metrics. What I am saying, and I am not saying for ICRISAT scientist or for anyone, you know that some people they work for an organization, something happens, points got exhausted, scientists were fired or whatever happens. If they would like to go to the new organization, the first thing in your CV they would like to see how good scientist you are, and the quick method is to see your publication. So what I am saying, so then this becomes an obligation to each of us in the context of institute, in context of taking care of those requirements of the funding agency as well as you. And some people, they say that for scientists, and they use this thing, publish or perish. So either you publish, otherwise if you will not publish, after 5 years, 10 years, the results which you produce, they will become obsolete. Someone else will do that one. And science does not wait. So you should not think that, well, I have constructed a genetic map for sorghum, let me wait, I will do this thing 2 years back. Guys, there are many other people working on sorghum or chickpeas or something, so no one will wait for you. So basically, if you are not publishing this work, if this is available at your desktop or laptop, then in my opinion, this is also not good for the organization because you have not shared those results. So in my opinion, this is really good. So that's the point I wrote, why we should publish. Next question is, where we should publish. So now the first important point is that as a scientist, we know that, well, what is our area or in which area majority of our stakeholders or are having access to that information. So this is the important thing. Nowadays, what is happening, majority of us are targeting highest impact factor journal. I'm not saying we should not, but in my opinion, each of us should try to see how many people, like I am working for instance in the area of genetics plant breeding. Majority of my stakeholders, they are reading the paper theoretical applied genetics or PNAS or genetics of plant breeding or UFIDCA, where I can target large number of audience who are interested in my work. How do we know this thing? This depends that when you are reading the papers, then based on this thing that, well, in this journal, this paper was published, etc. So this is one. Second is, this is again my personal views and I'm not representing right now management view or so, but in my opinion, I'm the big supporter of the open access publication. So now many journals you publish, but then somebody can search your abstract, but when they would like to download, they say you need to pay $45 or $65 for getting this paper download it. So, but if your paper is open access, then basically this is truly international public good. This can be downloaded by anyone, and then you are having. And right now our institute, ICRISAT, is a big supporter of this open access policy, and so you might have heard few weeks back that we had this, what was it, Open Data Week or Open Access Week? Yeah, Open Access Week, which was organized by Dilip Kumar. So this is very important. But in the name of open access, nowadays there are large number of journals, and some people call them predatory journal. Predatory journal, so yeah. So basically they are money making journals, they don't have any value. Majority of journals, they are coming with due respect to the different countries, China, India, Iran, or many different, even they write their names because they think if you will say I'm coming from US or UK, then I will be recognized. They put some name like John Milton or those kind of things, but they are basically rubbish journals. As a scientist, this will be you should know that, well, this journal is rubbish or this journal is good. I'm not saying you should always publish in the journals which are impact factor oriented, but this doesn't harm you if you can target the good journals. I'm talking we should not publish when we have invested a lot of resources, time of large number of people, and then we should not go in those rubbish journals. So this is very important. We have library, many of us, especially for the young scientists, you are having your mentors, your supervisor. When you are publishing, you can also ask your supervisor whether I should go there. Similarly, nowadays there are many conferences. Every day I receive several mails that we are organizing this conference, we have selected you for this one, etc. Again, they are not really genuine conferences. So you need to see that where you are working. This is important, it's not important, but especially for the young generation who are doing their PhDs or postdocs, that in which journal they should publish, and they always want to publish in high impact factor journal. That's okay, I'm not discouraging you, but they say that the policy is that you should try to identify your area of research. If you are working in genetics, based on your experience, you will know that my research is reaching to what level? Science, nature, or for instance PNAS, plant cell, plant physiology, tag, plant breeding, etc. So you can do that one. Now the question is whether you got some exciting result, you should directly jump to science, or you realize that, well, my research is coming in the range of that science of PNAS of plant cell, then maybe I can try PNAS. If this doesn't work, I can go to plant cell or genome research of plant physiology. So you can play in that area. Or if your research is just for plant breeding, tag, etc., then you should not try to say, okay, let me try in that one. So you as a scientist should we able to identify the boundaries of areas where you should go. And I don't mind that if you want to try in tag first, and if tag rejects, you can go to the plant breeding or you fight, so you can do that one. But at the same time, I will also not suggest you that if you know that your research is more breeding oriented and you started to publish this thing in some cell biology oriented journal, that of course in the first day they will say this result does not fit to the theme of our journal. So this is very important. Okay. So let's keep on saying what we should do for experiments. So this is another important thing. Sometimes this becomes difficult. So what happens that if you are very systematic and you plan, I would like to achieve this one. What kind of experiment I should do? You can plan. Majority of time what happens that we do some work, we plan something, we get some different kind of results, and then we start to publish that one, what we should do. But at the same time, in any way when you are doing your work, in my opinion, we should not be casual when you are planning with the experiments. You should spend a lot of your time in planning. Because once you plan your experiment, then you cannot come back, oh my God, it would have been nice if I should have used this one or if I should have used this one. So I would suggest you should spend your quality time in planning your experiments, including all kind of possibilities that you need to use control, etc. Then you are having the next component, execution. You should be very systematic in executing those planned work. This is very important thing. Data recording, data analysis, statistical analysis, all these things should be very sound. Many times people say that majority of us or all of us, and if you say this in terms of statistics also, we are very good in executing the experiments, but not that great in planning the experiment, or even if we can do, we are not wonderful or great in this data analysis. The same data set, if you give to two different people, one set of the people or some people, they can do fantastic analysis, they can have great interpretation, even they can give the different kind of theory. On the other hand, the other person, they will just say 2-3 lines about this data set. And for the high quality paper, your data analysis, interpretation, making some theory or confirming those hypotheses, very, very important. As I told you earlier, in my opinion, we should be very nice, we should spend a lot of time in planning experiment and data analysis, making interpretation, etc. So this is very important. We should not be very casual. Anyway, what else? I have developed a map of the 500 markers, and 500 markers is good, but the next question is, what else you can do with this genetic map? Try to do that. So those things are very important. So don't worry that, well, you have generated this data. Nowadays, anyone can generate the data. Question is how to convert this data in the information or in the knowledge. So this is much more important. Just generating data doesn't mean that you have done each and everything. So this is very important. That's the reason. In fact, your authorship, corresponding authorship, first authorship, secondary, tertiary, etc. also depend on your contribution in terms of the analysis, interpretation, etc. Not that just you generated data, because technician can also generate the data. Data is being generated by machines if you are doing the genomics research. So this is not great. As a scientist, we should spend our time in data analysis, interpretation. This is much more important, and we need to be smart in that one. Next thing is never cook the data. Sometimes you know that, well, this is my hypothesis and I need to have this result. If your supervisor is busy or traveling here and there and you thought, well, he or she may not have that much time, let me do this thing. Guys, this is the end of your career. Never ever compromise with this kind of thing. Cooking data is not acceptable at all. The day when your supervisor or so, they come to know, then this will be, and I know several examples where even some laboratories were closed down when they investigated that this data was fabricated. So this is not acceptable at all. In the name of publication, we should not, okay, sometimes you may have the less amount of data, sometimes there's different story that you are not able to prepare or develop your hypothesis, etc. But data should not be, we should not be playing with the data set. Because never report wrong based on this cooking data because of being impressed with the current trend of publication, etc. While they did this thing, my data should also tell this thing. In my opinion, this is not acceptable. You should always report what you observe. Don't need to be worried that, well, that laboratory A from South Korea or from China or from United States, they reported like that one, I also need to do that. This is my suggestion. Okay, so this was about that one. In my opinion, this data generation, how we should analyze, how we should interpret. Now the next thing is, and I'm giving this example based on my experience with my colleagues working with me from their PhD or MSc time. So what happens is when we read the paper, the first thing is abstract, then we have the introduction. So my colleagues also start to write the abstract first, then introduction like that one. In my opinion, this should not be the way. When you complete your experiment, the first thing is you should come up with the tables and the figures. Your results should be summarized in the form of the tables and the figures, which should be like self-explanatory. Then you can sit with your supervisor that I have got this interesting information. Based on those discussions, then you can give the orientation or direction to your results or to your work that, well, these data indicate this one, and the direction or the orientation of my paper should be in this direction. So basically, I'm one of those guys that I should discuss or prepare my results and try to interpret them first, not writing introduction or material methods, etc. In fact, in our lab, when I'm trying to do this thing, this doesn't mean again that we are the best. What I'm saying, we are just sharing our experience. So what I'm saying, that in my opinion, abstract should be written in the last. Introduction you can write later stage, because seeing that what kind of results, etc. you are having. But don't start to write abstract and then introduction in this thing. This is my feeling. So first is you should know that what you want to do and what is happening. So, and nowadays, and this material methods is very important, but you should write in the last, in my opinion, because that, well, you should see what you have reported, because whether you are covering each and everything, material methods. Nowadays, many high quality journals even, they also, you might have seen that they put material methods in the last. Earlier time, many journals, they used to have this material methods in between. Now even people like Nature and Science, they are having every material method as a supplementary table. They are having just 2-3 paras where you can write that what is the main thing, but then they are putting in the last. So this is my way of thinking. Don't start with the abstract introduction, etc. Rather, start with those results, interpret them, etc. Okay, maybe we can discuss. Some of you may not like this idea, but anyway, let's discuss this thing, right? Okay, good. Now, as I told, so many of us, especially young generation scientists, they are interested in high impact factor journal. From our lab, we know. So sometimes people say, hey Rajeev, we try on this journal. I say, well, but this doesn't fit there. Why we need to try? So sometimes they think I'm discouraging them. No, this is not the point. Point is, we need to realize that which journal fits better to your results. So this is most important. But anyway, if you are having, and sometimes people like to say Science, Nature, etc. and based on the discussions with different editors or some of our experience. If you work hard and then you prepare a manuscript, submit to Science or Nature or like that one, and if this doesn't fit in their journal, they will send next day response to you, because in Nature there are professional team working in United States and UK, and they are very smart people. They don't have that editors from the scientific community, they have the professional editor. 158 editors for the Nature groups. So they work day and night. They just quickly analyze these results, see and say, well, this doesn't fit. Sometimes they discuss with their fellow editors and say, do you think that this result fit to your journal, etc. That's okay. But what is happening nowadays, that they are having, in many journals, they are having this pre-submission inquiry. So you can just share crisp summary of your work, 1 or 2 figures or tables, etc. and then based on your inquiry, journals can tell you this work fits to our theme or not, so that you should not work hard or you should not waste a lot of time. So you can do this thing, but when you are preparing the summary or when you are interacting with that one, you need to highlight what is unique thing in your work. Don't say this thing that, well, in the case of chickpea, this is first time because other people did in rice, wheat or vigna, mungo, etc. So this doesn't work, that this is, in my crop, this is the first time. Many times we say, nobody has identified markers for fertility restorer in pigeon pea. I'm the first person that I'm doing identify. This is good for making yourself happy. But then fertility restorer is not new thing, something like that. So you need to see. Of course, this is important in the pigeon pea. Then you should target the pigeon pea or legume or breeding oriented journals. Okay. So then, and the other thing is that if editor responds positively, then they start to prepare the manuscript, etc. For instance, in the case of PNAS is also very high quality journal. Many of you may be aware. This is the Proceedings of National Academy of Sciences. They have the different system. When you submit your paper, for each paper they have an editorial board and they are assessed by 6 members. 3 members from the editorial board and 3 members, they are the member of National Academy of Sciences, and you need to suggest these names. 6 people, they see your paper, then they realize, they assess and they decide whether this paper should go for review or not. So I mean those kind of things. So different journals, they are having this thing. But another thing is, and yeah, while you are doing these things, we know that especially people coming from India, I know that including myself, we have the serious problem of the English language. If we think sometimes that we have written very nice English, but honestly speaking, when I talk to my fellow colleagues or say, they say, Rajeev, in fact, this is not what you are talking. This written English is different what you are having in your mind. Many times we think that I have written what I wanted to write, but this is not the case. This is one of the serious problems with many of us, and honestly speaking, I don't know what is the solution. Maybe one solution is that when you are working with your colleagues, and some of them, they are native English speaker, talk to them. Or nowadays there are several times these editorial services, you can check with them that well whether they would like to help you. Otherwise you have done wonderful results, and because of your language. But many journals, high impact factor journals, they say we don't reject the manuscript because of the language, because they also have this copy editing service, etc. So this is important component. We need to see what we can do. This is also important thing. Many times we cannot present our results in that concise manner. For saying something we need 4 sentences. If you go to the native English speaker, they can tell the same thing in the 1 sentence. So, but again, this is something which we cannot change. We cannot start our primary education from those English medium or so. So anyway, so this is the thing. Now, next thing is that, well, I already told, always write what you think is correct, and if required, you can ask your English language friend that you want to write this one. And I am also having the same problem. I am telling you this, accepting. Many times, and I am blessed and I am very thankful to many of my collaborators and friends, they tell, hey Rajeev, this is not what we were discussing, this is different. I say, yeah, I am sorry, can you please help me? I don't mind to take the help from those people also. So we should, and many times we should not be just carried away by some sentences from some other papers. That well, they wrote this thing and if I write this thing, because when you, whatever you are writing, this is my feeling, you should read and try to see that, well, at least, or what you understand from that sentence. Many times we feel that I am understanding this one, but if you read 2-3 times, you will realize this is not correct. So this is very important. And I said that, yeah, many times MS is rejected, which should not be the case, and I had several times arguments with several editors that they have rejected the manuscript from our group itself in the first instance. And sometimes, I should not mention, sometimes these editors, they are so much biased or prejudiced that if they see that manuscript is coming from India, and even if I have got this manuscript checked by some native English speaker, just they see corresponding author Rajeev Varshney and coming from at India, they say, it will be good if you get your manuscript. I say, well guys, I already have done it. And this has happened 2-3 times. But anyway, we cannot help. So sometimes I had the argument as well that, well, and I say, again, I am not saying that you should write in the bad English, because in some cases I was convinced, that's the reason I did. I said, I am not writing an English novel or some English story for targeting in UK or Australia or United States. I am writing a research paper which should be read or understood by large number of the people. Tell me which sentences are wrong. I would be very happy to understand. I would not like to listen this thing in just one day and you are saying that language is not good. Okay, so these are very important things. So what I said that, well, it will be good to have the people from this thing, so this is important. So this is important thing. This is very, very important, and I am having serious problem even with my colleagues, and although our lab publishes large number of papers, but at the same time many papers, they are not going out, because I am not happy, because I cannot be convinced that, well, these people have not copied and pasted the sentences from the other paper. This is a big problem majority of time, and this is for young scientists and also in my opinion for senior people. So guys, just for the sake of your career, don't play that you just, nowadays internet days, you just copy, paste it in your paper. You think that your supervisor will not realize. Your supervisor may not realize, but when you will send the manuscript, and if you are targeting some journals, nowadays many journals, they have this plagiarism tool and they can just put your text, they can figure it out that 20% or 30% of your language is coming from already published. Even you should not try to copy and paste your own paper. So this is also important. So we should always be very much smart at that one. And this is, I saw that what is this dictionary meaning, that it is the wrongful appropriation and the stealing and publication of another author's language, thoughts, ideas or expression. We should not work in that. Sometimes, as I told, English is not our mother tongue, but at the same time, if you are young or if you are just writing your first or second paper, some of you may be knowing, I ask, when I realize this sentence looks great, then I start to have the feeling that this is not written by you. I say, well, tell me from which paper you copied this one. And I always tell me that this section you have copied or something, I will try my best to modify in my own language. But don't use this copy and pasting. This is a serious issue. So these are the several problems that why our papers are rejected or we are not able to publish this thing. Anyway, this is I have learnt, this is my experience. My English always gets spoiled or bad when I start to write the longer sentences. So I feel that, well, writing longer sentences is good, this indicates that your English is good. But when I start to do that one, and when I ask some people, they say, this is bad, this is not the good synthesis. So I feel I try to write the smaller sentences, but still I'm not perfect. This is the problem. Many times coming from Indian background, and I think, I don't know that our colleagues from the Africa, but they also have, but from people like Kenya or so, which have been English colonies, I think their English may be better. But I know from my side that earlier I used to have this tendency to write lot of stuff in the parenthesis, comma or parenthesis like that. They say you should not try to put these things. So should try to avoid these parentheses to tell your story. Okay, this is another problem, and I am having this problem, very serious problem, that people they put the table and figure, but in my opinion, your tables and figures should be self-explanatory. In my opinion, these are the souls of your paper, that if someone reads your table and figure, they do not need to even read your text and they should be able to understand. Sometimes I start to say, well, but what I should do? But if you see the other papers, etc., they have the footnote, legend, etc., very long. Because idea is your table and figure should be self-explanatory. So you need to do that one. This is another thing, high resolution figures. When I come to you, again I am telling my experience, my colleagues say, if I will prepare 600 DPI, figure will be this big, this will not come in the 2 pages. I said, this is not your problem, we can put on the HTML, this is journal's problem. You upload that figure, we will see. In my group I am having this another problem, that even figure is big, the font size will be 7 or 8 that you cannot see by your eyes. The question is, why you need to do these things? Why and I am telling again, so when nowadays in computer era, I always tell my colleagues, you see in the 100%, if your eyes can read that thing in 100% zoom, this is great. But you should not go very close to your computer when you are having 100% that what is written there. You should not be worried that size, etc. You should tell what is that. And also don't try to use all these possible colors, violet, pink, yellow kind of color, which doesn't make any sense. Try to use first primary color, then come to the secondary color. My colleagues majority of the time they start their tables of figures with the tertiary colors. This is not good. That's the way they call primary color, secondary color, tertiary color. You also prefer figures in pink and yellowish color or what? Turquoise, will you? So do not worry about the size of the figure in my opinion. Okay. Now, what is that? Yes. So another problem. In my opinion, you keep on telling a lot of stuff, but whatever you write in your paper, in my opinion, should be substantiated by your results. Sometimes papers may have this problem of the size. You can prepare supplementary tables, figures. Many journals nowadays like it. If journals is not taking, in my opinion, many times you can see the CEG website, they are also putting the supplementary material on those websites. This is very helpful. First, to convince the reviewer. Second is, even if you are sharing all these information in the public domain. Okay, another thing is that, again, I am telling my experience. So when I work with my colleagues, they bring sometimes in one paper 170 references. They are having the references even 1980s or 1945. This is not the way. Generally, they say that if you are having a good paper, they should have average 40 references. This is good. And even when you are having the 40 references, my feeling is that 50 to 60% references can be from your last 5 years. 30% references can be, or if you can add 5 more years, so maybe the total 10 years. If you are having the references old, maybe not more than 10% references should come from the 15 years. Unless this paper is very great or legendary paper. Now, for instance, all of us work on DNA. Do we cite the paper of Watson and Crick? No, there is no need. Similarly, now RFLPs, now I was one of them when I was doing PhD. Every time I was having Botstein et al. 1991 RFLP math paper. What's the need now in the present era? So we should see this thing. If you will continue to cite your old references, as a reviewer I will feel, well, either this person doesn't know or his or her research belongs to those 1960s, not in the current era. So this is another thing in my opinion. Okay, now once your paper is done, and again, so this is just experience, this is not the perfect solution. So I have not taken any class or I have not picked up from the internet. I am just sharing the experience, which may be bad or which may not be. So then, in my opinion, that then we should have the cover letter. What I have seen that when I tell my colleagues, and this is also my duty to train this next generation of scientists, that's the reason that I thought, let me share this thing with you guys. So what happens when you start to write the cover letter? Again, you copy the whole abstract and paste in this one. Try to avoid that one. Otherwise, editor will read that one and then this is also the same. Of course, in abstract provides a lot of information. Try to take the unique points that, well, why you need to have this problem. And sometimes nowadays this is happening, the problem with our in the China or India, etc. So when they are suggesting the name, and I am telling this thing, journals, non-Indian and non-Chinese journals, if you give the names of more than one Chinese or Indian editor and reviewer, they can guess that, well, your work is not of high quality. This is the problem that people start to have the feeling. So I say, well, why you are giving this opportunity? If your paper is not published in that journal, doesn't matter, you can publish in some other journal. But don't get your paper rejected in the first instance that, well, your cover letter is not here. Another thing is, avoid to give your friend's name as a reviewer. Many times we feel, if I will give the name, because they ask for suggested reviewers. Sometimes when you give a lot of things, then people, I can, as an associate editor for several journals, when I read these things, I can easily pick out that, well, why this person is giving this name. They say that you should not give the name of that collaborator friend who has worked or published with you for last 5 years or so. Editors are that smart. So even if you will give, they will use this information to populate their databases, not to send your papers to that particular reviewer. So anyway, you are working for journal, so thanks for that. But because another problem is, and this happens with some of our papers as well, coincidentally or so, these papers, they kept on going to one particular editor. Your work is of high quality, but when you will be publishing this thing, people will think, oh, Rajeev's friend, he is publishing this paper like that one. So I feel embarrassed, not other people, but so this is not good, that why we should do that one. Okay, so then they go for review, etc. So this is my feeling, and I'm going through the whole pipeline. Now, when we receive the feedback, you can have only 3 types of feedback, that people say your paper is good, accepted. But nowadays, those days are gone that people were accepting paper in the first revision. They always ask for suggestion, etc. Sometimes your paper can be rejected. In my opinion, if the journal has asked revision, start to work on the revision, but you can send a letter to editor. Don't think that, well, they have asked for me 2 months, etc. Sometimes you forget, editor forgets. It's always good to send a thanking letter to editor. Thank you for rejecting our manuscript. Yesterday itself we got a rejection on our paper, right? So I also send a thanks mail to him or her. Thank you very much for considering our paper in the journal. This isn't hard. I mean, just sending this thing. You should not take it personal. Oh my God, that this reviewer has rejected your paper. This is science. We should not always think that all of our paper will be accepted in the first instance. I'm one of those guys, and don't feel insulted or like disappointed, discouraged if your paper is rejected. Paper is rejected because of 2-3 things: that the journal which you targeted, this doesn't fit to that theme, or that area is quite high, etc. Maybe second is there's some problem with your experiment. Reviewers need to be convinced you did not do the good analysis. You can do those revision. Sometimes they want more data, so you can do this. I don't need to feel disturbed or like these things. So this is okay. This is life. Okay. Now, when we receive the feedback and then they say that, well, you do the revision. Generally, I do this thing that when we are having the paper and we are having 4 or 5 different topics, at that time itself, you just ask your collaborators, colleagues, guys, can you take care of this part? Can you take care of this part? Instead that you start to think, I will do everything. I suggest at that time point, you can plan and then send a message to your collaborators. I have done this thing many times. When I am having the bullet points, I told, guys, can you take care of these 3 points? I will take care of these 4 points. You take care of these 3 points. This makes life easier and our revision will be faster. Okay. Well, I wrote this thing. This is our plan that first author should prepare. That's what we do, that either first author or corresponding author should prepare the responses to the comments, and then they start to revise the manuscript that, well, let's do this thing, let's do this thing. Instead of asking 4th or 5th number of author which doesn't have any clue, so I would suggest the first or corresponding author should take this thing. Never criticize the reviewers unless they are very stupid. Sometimes reviewers can be stupid as well, so that's not a problem. But when they don't understand your point, this is our duty also to tell them that no. So, but generally we don't need to, but sometimes my colleagues know that I have scolded reviewers also, editor also, and then they understood, then they got back that, well, sorry, this was not the thing. So this happens, but I'm saying in general, we should be nice to them, otherwise they will not be nice to us. And then when you are submitting your manuscript, because you have to prepare this cover letter again. In this cover letter, you need to ensure to highlight that you have revised the manuscript. Don't say that for everything, yes, I have taken care of this suggestion, this thing has been taken care, this has been taken care. You need to provide. Where did you do that one? If they say that, did you do the analysis? I say, yeah, I have given the analysis, this is presented number in table number 5. Don't say this thing, I have taken care of that one. I am having this struggle with all of you. I don't want that you should say, yes, I have implemented suggestion. I would like to see where you have implemented suggestion. And when you are revising the manuscript, always use track change command. Okay, so these are the ways. And then, yeah, you should have this file separate response to comments. And you need to prepare, answer very correctly, very clearly what. If you are not agreeing, you should tell, I don't agree with that one, but you need to do that. Then you can submit this thing. Okay, what we should do when we receive acceptance of your manuscript, feel very happy, of course. You can send in your mail to your boss to make him or her happy, and of course co-authors, not to all your friends on the Facebook at that time point that my paper got accepted, because you never know, this can be rejected again when they realize that they did this, and then have celebration, that's okay. But then your job is still not finished. The most important thing, and many times we have this problem here also, that when you have these galley proofs, what happened that when you were revising the manuscript, this happens with me once or twice, and I feel very, very disgusted. I thought that first author should have taken care of this thing, and I did not have time at that particular point, I am admitting. While revising this manuscript, added more problem. And then when I received the proof, I said, my God, what happened? And when I checked back, I realized those errors have been introduced at the time of the revision. So this is the last stage, and this stage in my lab, I really want to take it very seriously. And this happened in 1 or 2 cases, I know, and where I told editor and I had to ask 2 times corrected proof. So I don't want to leave, and even sometimes these cases still happen. So this is the problem. The reason when this +1 paper in the peanut, that these people, they just changed the legend from figure 3 to figure 4. And I felt so bad, but now they have corrected, I mean, this thing. So this takes a lot of time, but these are sometimes very small things. So spend your quality time, and in my lab I always say, all co-authors give everything. And sometimes I'm having this phenomena to motivate or something. If you will find really 5 good mistakes, I will give you 5 chocolates, 1 chocolate for each good mistake, or something like that. Or I will offer you the tea, or I will take you for lunch. So people feel motivated, let me find the mistake. I always feel happy when someone tells my mistake so that I have an opportunity to improve myself. If you will not tell the mistake, but these things should be constructive. This is always my feeling. Constructive criticism is always good. We should not just keep on criticizing the people just for fun. But, and this is my suggestion to all of you, including myself. You should not try to be defensive that if someone told, hey, you did not have this figure, this then, and say, oh, why like this thing, I know more than you. I mean, no, this is not the case. So then send this proof to several colleagues and then send edited files of proofs in 24 to, then when paper becomes available, then you can have the real celebration. At that time you can tweet your paper or you can share this thing on the Facebook, etc. Okay. So I think that's what I'm having. Thank you very much for your kind attention. I would be very happy to have your feedback on this one. Before that, can I have my assessment from you? How many of you liked this presentation or you thought that this was useful then or you think that this was boring presentation like other presentation or like this thing? So can you please raise your hands that this was really useful or you think that this was just wastage of time? Thank you very much. So I think that this is good. Our colleagues, they are also there on that online? No one is there? Or went, okay. Okay. Oh, good. Okay, good. Anyway, so we can have the discussion and we can like to have your feedback because you can add something to share with the group that no, this was rubbish, rather we should do like that one so that this will be helpful to the group. Who raised the hand for adding the suggestion? Noel, you would like to add something at this time point? Noel, Julie or someone else? Yeah.
Thanks, Rajeev. I think it was very helpful to do that, and I think it's certainly it's good to spend time thinking about these things and work out what you might do. I'd just like to make a comment on what you said about English language, and said it's difficult when English is your second language. Like I've had a lot to do with people for whom English is their first language and people for whom English is a second language. And quite often, actually, people for whom English is a first language make a real mess of it. and people who have English as a second language do better, because actually they've thought about it quite hard on how to do it, whereas if it's your native language, you just you jumble the way you present things. And I guess in a scientific paper, you're really seeking to try and be exceptionally clear. Like if you're writing poetry, you're not seeking to be clear. You're trying to make allusions and make people think about other things. And in natural language, you might be trying to make fun or amuse people by the way you say things. So your language is much more layered. And in a scientific paper, language is pretty brutal. I remember once actually being at a talk where Maurice Wilkins spoke, and he gave a description of science that has stuck with me. He said it is intellectual thuggery. So,
wow. Thank you very much, Noel. And as we know that Noel has published many high quality papers or so this is getting those kind of feedback from him is really good. Julie, you had some other suggestions?
So
you know trick how to speak on this.
we were in this room last week talking about presentations, and one of the main points from that was when you're giving a presentation. This is not about your presentation, by the way, but it was if you were able to distill it down to 3 things about writing a essay are the 3 most important things. I think that's what we need to get to. I mean, you many things to think about there, but really I think we need to get it down to the crucial what's the point of a paper. So what would you come down to?
Okay, so now you are asking the question to me, right? Okay, I thought that you are adding something. No, I think by the way, as you said, so first is, another thing is, so I I went through the whole pipeline of writing the paper. So basically the idea was not just to give the script form, but this is very good point. Second is the presentation which I have made. By anyway, I know that this was not the good way to make the presentation. I did not have any figure, I did not have the table, so don't worry about this presentation. Coming back to this question, that what should be the way? In my opinion, and this is my feeling, the first thing is data analysis and interpretation is the most important. That how you have analyzed, how you have interpreted results. This is my first thing in my opinion. So otherwise, I have seen several people, many of my collaborators, and I always, and during last, in my opinion, I am publishing paper from 97 or so, so now this is more than 17 years. I have learnt a lot, I am still learning, and I always feel really surprised that, well, with the same data set, I could not get those kind of very clarity, those results interpretation when my other friend, for instance, Duck Cook or someone Chuan Chang Liang in China or Scott Jackson or Gunter Karl, they made fantastic this thing. Why could not I? And this depends on data analysis. So I have learnt a lot from that. I always say, what the heck you could do this thing and not me? So this is very, very important. So this is one. Second is, in my opinion, that when you are presenting something, this is my feeling, and I'm talking in terms of the publication of the paper. If we are able to convince the reviewer that whatever sentence I'm writing and I'm having some backup thing for that well, or substantiated thing, either in the form of the tables, figure or so, I'm not just talking in the air. What I'm talking, this is what I observed. So this is my second thing. I don't know that I need to go for third or fourth, but this is the thing. And of course, if you are having this thing, this is the, I still believe that high quality figures really very important. You would like to add something?
Yeah, I would also say start with a question, start with a hypothesis.
Yeah. Okay.
And so there's all this business about English style, English language. It's a little bit secondary.
Yeah. Language is not that, that's the reason I did not put language there. So, but what I told, so I did not put this hypothesis so because I already assumed that you got the data. So after that I was starting. But when you are having something in your mind, you would like to test your hypothesis, of course you should have your planning of experiments and hypothesis very clear. Good. Thanks for this very useful suggestion. Okay. Any other suggestion also? Manish, can you add this thing quickly? Because I will give very brief presentation on some other topic in case if you have some suggestion. Colleagues from DC? Colleagues from non-DC and non-grain legumes?
I have another comment, if I may.
Yeah.
This publish or perish thing.
Yeah.
It's a it's a gripping kind of statement.
Yeah.
And publish or perish. I think even when you publish, you can perish. It's not a dichotomy. It's not a dichotomy, right?
You should be sustainable publisher.
So it's about quality, right?
So yeah, the other thing is, so this is another thing. So many times, and that this is good. So a few things I would like to add. So that these papers, the journals, right now you are having the range of the journals. In the scientific community, we are also having range of the scientists. We should always try to come in the elite class, no doubt about that. And if I'm seeing just quantitative variation, so you will have only not quantitative, but let me say like that. So there are only few people who are very elite scientists. They are publishing high quality work, etc. For them, they have this thing where there is what you call bar of or standard bar somewhere, yeah, benchmark that if you will have the data below of this quality, they say even I don't care for publish, I will publish only when I am having the data for the journal like plant cell. I don't care for this tag or plant breeding or plant genetic resources characterization and utilization. There are those kind of scientists. Great. Somehow, and then, but I'm not saying this. So the point is that we should understand the organization where you are working, the area where you are working, the objective for which you are working. Now, if you implement these things in the context of the CGIAR system, as I told earlier that CGIAR system or research, which I learned, I may be wrong, that our main job is not to publish the paper. Our main job is not to publish in plant cell or PNAS. That's not our main job. That's the reason I was trying to say this. Now, My thing is, if you have generated some information, you know that this cannot be published in the plant physiology. So now the question is, first is, I'm not working in the area of genetics area or PNAS research area or plant cell or cell biology, etc. Now the question is whether I should publish. I'm a big supporter that whatever you have generated, you should publish. Because my opinion is not to have 200 papers in my CV. My main purpose is right now to share with the community what I have generated. That's the reason if you see from our lab also majority of time our papers are open access. I'm the big supporter. So not only this publish, put in the open access. So this is the way. But when I said that you can publish in plant genetic resources and characterization of plant breeding or you fight or any journal, but not at the cost of the quality of work. Your data set can be smaller, your area can be narrow that you are targeting only this thing, but this doesn't mean that you should publish wrong results. I don't want to publish in a journal which does not have impact factor but high quality journal which is going to large number of people, right? But at the same time, if I start to have the feeling, and sometimes in many universities this happen, and what happens, who is the sufferer for this problem? I can tell you, I know many people in China and United States, these laboratories, they publish the paper in Science and Nature or Plant Cell. Now in these labs, they are filled with 20, 30 PhD students, postdocs, they are having publication once in 2 years or after 3 years, etc. Great publication. What happens, I am not saying, but now this thing is different. Many of the PhD students, they don't get publication, a single publication even after 7 years. They can say, I don't care, but I don't know. So this is the difference. In my opinion, I am having due great respect for those people as well. Unfortunately, in our system, or at least in our lab, we don't have that until unless we have a paper for the genetics or tag or something, we should not publish. I say, well, field crop research, tag, plant breeding, anything. Because our main purpose is to help those young scientists who have done their work for last 4 or 5 years. If I will start to do that work, they will be perished. And this is also an obligation to us, for those people, and also for the funding agency, for the organization, etc. So this is and this varies from vary to vary. Yeah, so Julie, just a minute, so last point which I wanted to make, yeah, I will come back to you, about this, that you can publish but it still can be perished, yes. And I know that in the National Academy of Science in the United States, when you want to be elected in that academy, while talking with several people who are in those boards, they say this is not important that you publish 3 papers in science 10 years back. The point is what manner you have impacted the quality of science, how much sustainable you were. This is not that you publish 1 paper in Nature Biotech and after that you become a hero, that well, you have achieved each and everything. This is also very important. So this sustainability is also very important. Okay, so Julie, share something quickly and then we will go ahead for next. You are having
Well, I would agree that high quality can come in any level of journal. Plant breeding, tag, any level, quality can come.
Yeah. Good. So very good. So now let me, okay. So we will use just 10 minutes to, it's not coming, then you can have the old computer. We need, okay. What I thought that I wanted to, thank you very much guys for your patience, and let's, this thing. So Manish, why don't you use the old computer? Why you have wanted to use the new computer? Okay. So now this is for all the colleagues, because sometimes some of us are having the feeling, and I thought to have this disclaimer. So, okay, can you please take seats, or Lekha, you don't want to listen us now, or because somehow, you know that, and this is not that I am defending myself, so don't think like that, but the point is my major job is, and this was one of the important point of the critical focus area of molecular breeding, CFA, that even the breeders of the so the point is that this center of excellence in genomics, although we talk about the genomics research, but the main purpose was that modern science, and here we can say genomics for instance, should be the integral part in the different disciplines. Sometimes if you are working in some other building and you say, oh, genomics means that only Rajeev Varshney or Rajeev Varshney's people can use, no one can else do this thing. This is wrong assumption. So I want to tell guys, you should not have that assumption. If you just keep on having the assumption, you will not come out, then you will make your theory confirmed. So when we have this genomic center, this does not belong to grain legumes only, and of course not at all to Rajeev Varshney. This is institutional center. This goes across the research program, across the locations. And we would like to see that all the different disciplines, all scientists of different age groups as well as of the different cadres, they should be making the best use of these facilities. Sometimes these facilities can be, light ko band karo, sometimes these can be that busy, but then we always have this thing. So right now, very quickly, so we are having those 5-6 facilities at the center. One is this SSR genotyping, SNP genotyping, but SNP genotyping is being phased out now because of several reasons. We are having MySeq facilities, DART genotyping, this will also be phasing out. Now we are having this SNP genotyping by using Casper markers, and we have HiSeq 2500. So these are the machines, or of course you have the QRT-PCR, the basic facilities, etc. But these are some of the core facilities what we have. The objective of this Center of Excellence in Genomics is not just advancing grain legumes research. This is for the ICRISAT. I am highlighting again, this is for ICRISAT across the crops, across the disciplines, across the locations. Sometimes what happens, this is the way, that this depends that, well, how many papers you are publishing grain legumes versus dryland cereals versus non-crops. Until unless you have the project, until unless you do the research, etc., how you think that these papers, so you should, sometimes you could say that Rajeev is having, sometimes in jokes also I say, they say, how you are publishing so many papers? I say, we are having a printing machine. We put something and then we ask, this time I want paper in tech, this time in plant biotech journal, this time in PLOS biology, machine gives. So don't think that I am having the machine and we are just putting the data and coming like that one. This is a direct function of number of people, number of the grants, number of your collaborators, number of your ideas, number of hours you invest. If you are missing one of these things, this will directly impact. Again, as I told earlier, guys, objective of center is not to publish the paper. Objective is center is to introduce, to infuse the modern science, the genomics research in the different disciplines. Now, if you are not aware, and Anu, you should take these notes and then you should circulate these things. So facilities, being the director, I am responsible. Anu Chitkineni is the manager. Her role is not to dominate scientists. Her role is to work with the scientists to get the information. So whenever you are having any projects, you need to contact her, so that, because in her role as a manager, she needs to manage those different operations. Why? You come to me, SK, I want to sequence 10 lines. Now this will be role of Anu, that she needs to see whether she got chemicals consumable for 10 lines additional or not. If not, she will say, okay, let me order the chemicals and we will come back to you in 5 days later, 10 days later. So this is the way things happen. So anyway, so then we will come back to that point. So now what will happen, those interested scientists need to contact manager. Plus, if you are working on chickpea, it will not hurt you if you talk to me, or if you are working on sorghum, you can talk Santosh or Palminator Rakesh or anyone, that we are working this thing. So you can talk to those concerned scientists, they can help you whether you are having primers for that crop or some particular thing which you are looking. So you need to do that one, but you need to make the best use of those facilities. They don't belong to one person, they don't belong to any group, they don't belong to any location. So this is very important. We have these facilities. Now, as I told that publication, so don't combine these two presentation, because the first presentation was on publication. So you should not have the feeling that Genomics Center objective is to publish the paper. So our objective is, as you can see, that we are having those sequencing platform, we are having the genotyping platform. The main thing is, you disappear something from here. So this was the text, that here this was that working with those genetic resources, physiologists, breeders or cell biologists or wide hybridization specialists. So basically work with those germplasm, prepare those genetic maps, collaborate with those different type of phenotyping, try to identify the QTLs of genes, use them in the breeding program. So this is our overall objective. And yeah, so now let me say, and okay, I also need to introduce again Mahinder. So Mahinder, where is Mahinder? Okay. Mahinder is heading the Genotyping Service Laboratory, which is one of the component of Center of Excellence in Genomics. And Mahinder, and what is the other guy? Vinay? Here, okay. So Mahinder, Vinay, Anu, these people, they take care of the different kind of experiments. Now in the center, what is happening that some of you grain legumes, because we are having many scientists in grain legumes, special project scientists, visiting scientists, they are already working there. And then we are having lot of people from the dryland cereals, Rakesh Srivastava, Santosh Deshpande, Chakradhar, they are working here. And you know that many of these people, I might have missed several names, but what I'm saying, these people are integral part of the center. If you have some question, raise your hand, or if I have written your name just to impress that audience, then also you can raise the name that no, you are talking wrong or you are telling lies, I'm not part. Because why I told that I'm listening this thing very frequently, people think, oh, no one else is working, only Rajeev is working. So this is one of these things that, well, all these people, they are at different level. Again, you can also come here, bring some money, bring some projects, bring some material, until unless now, how can, so, but this was for the genomics group. When I say genomics group, across the grain legume and dryland cereals. But on top of that, Mahendra has been providing the services to the, and I'm talking this thing only for 2014, this year. So these facilities are not only for ICRISAT, but even for the external partners. You can see that 16 organizations from 7 different countries, they have availed these services. My friends, when these services are being extended to the external partners, how you think that they are not available for you? Okay, so then, and this was the genomics group. Now come next slide. Okay, but not only genomics group, and this is the one of the highlight of these CFA meetings. Earlier only genomics people are working, but now you can see that for the other disciplines, people like Hari Kishan Shodri, who is working with Aspergillus fungus, sequencing of Aspergillus, resequencing. Mamta Sharma has developed the data array for that fusarium together with Mahendra and us. Her paper was highlighted in New York magazine that this was the first time data array. Now she is getting the best award or best, what do you call, outstanding scientific article. This was not possible earlier if we don't have the CFA meetings. She came, she says, I would like to do that one. Now she has sequenced the fusarium oxysporum genome. She is doing transcriptome analysis in collaboration with the scientists by using these facilities. She does not belong in hardcore the genomics group of that grain legume. She is pathologist, she is doing that thing. But if you are not aware that Mamta is doing, how can we help you, right? Now Srinivas Rao, he is not here, he is also doing some work on the sorghum transcriptome sequencing, etc. SK, you are doing genotyping by sequencing. By the way, these people have subsidized the cost of your project. This is not correct, so I will discuss sometimes with you. Anyway, so he is using the genotype. He is not hardcore genomics person. Rakesh is hardcore genomics person in Pulmonet. But as a breeder, he would like to do. He is working with Rakesh. He is coming to us. So I am giving this example that these people who were not working directly, Rajan Sharma together with Richard Michel Moore and etc. Rajan has developed the genome sequence. We are working the resequencing, etc. All these things, that I feel very proud that although we have invested a lot of time, and Anu, thanks to Anu that you have been organizing this meeting, etc. But the point is, by doing these meetings, we are trying to motivate colleagues. If you don't see your name, because Shivali is looking at me, don't worry Shivali, you need to come up. But anyway, this is a joke. But anyway, so then people who are coming from any area, they need to be highlighted here. Not only this one, we are having even collaboration with the RDS, Resilient Dryland System, where this one guy, Vasai guy, you are here now or not? Okay, he is not here. He is working with Suhaswani. He wants to do some work on Jatropa. He is working in the lab. So I don't know that why sometimes you have those kind of assumptions that, well, this is only for the grain legume, etc. Because now from these people you might have seen, well, this was external. Now this thing even internal, many people are already publishing the papers, working these things. So they are, now sometimes you may not be aware how to do that work. So we are having this kind of flow chart. We need to circulate this thing, Anu, to across the people. And I am always one of these things. So now if you got some new project, I would suggest to contact us in my role as a director, Anu maybe as a manager, and I also suggest if Mamta is doing something in fusarium related to chickpea, it will be good, and that's what I always try to do that one, to bring chickpea guy. If you come, I will associate with Rachit. If you come in the Pulmonet, I will seek advice from Rakesh. If you are working on sorghum, I will seek advice from Santosh, or if something is happening in functional genomics, Hemavindu, or like those kind of things. So basically we are having that you contact these people, discuss about the projects. We are not the company, okay? So sometimes you say you don't have this price list. We are not the company. We can prepare, but rather our job is not to make the money. Our job is to see genomics as integral part of your project. Money is not an issue, I can tell you. Many times I have told you, Shivali is one of them, that she says, I want to see heterozygosity test. No problem, come there. I never ask people that, well, this is the project, if you will share this money, then only we will do that. Our job is not done. We will, again in the end, this is Ikri said money. So we can work out, we can discuss that what is the size of the project, how much money you are having at the same size, and then Anu can discuss this thing that how much chemicals he got, and then Vinay, Mahendra, etc., how she should plan the experiment, etc. So then this happens. And when you are having this data generation, what happened at that time point, we can have this discussion again with these people. Many of you may be knowing that I sit with Rajan or with Santosh or with the different people, that well, we got the data, how to discuss this thing, how to analyze. In our center, we have a computational genomics group, 4 or 5 people, they provide the basic support. But then, so you can have this basic support from these people, and then after that, depending on their time, etc., then they can give you data back to you. By the way, during all this procedure, we don't ask that Rajeev Varshney needs to be the author or Rajeev Varshney needs to be the first author or corresponding author. Not at all. Our role is to help you, because in terms of authorship, I am already very happy to have large number of papers, so I don't care for those things. So never ever think that if you will come, we will dominate your research, if you have that feeling. But if someone, and then this depends to you, if you are a really good scientist, depending that how much contribution someone Krishna Mohan or I don't know, Mahendra or Rachit or Rakesh or Santosh, they have made, and you need to work with them. So this is the secondary scenario. But what I'm saying, in the whole scenario, this never comes. So if you are having this feeling or if you are having the fear, it's always good to have the open discussion instead of thinking in your mind. Right? So this is the way. So I would suggest that this is the way that things are happening in the lab, in the center. This center is open for each and everyone. We would like to see use of those facilities in your group or in your research activity as much as we can. Okay, I don't have any other slide after that. So with these things, you did not put the thanks slide. Thanks for the people. Anyway, so thank you very much for your cooperation, for your help. I would be happy to answer any question on this topic if you have. Any question from anyone? Any question from our colleagues from different locations? Someone is typing? Guys, this is your last chance to type your questions. Anyway, so I will be in Nairobi tomorrow onwards, so I can discuss with the colleagues in Nairobi if they would like to do something. But we will be very happy to answer. So I think that this flow chart is very clear that how to approach people. Can I request you to raise your hands that this is clear, or you still think that there is some ambiguity? How many? Rakesh, haath uthao yaar, jo hai pucha. So please raise your hands that this is clear. Now you are not raising your hand, so this looks ambiguity. Okay, good. Thank you very much for your time. You can have some social interaction if you want, and thanks a lot. Have a great day. Thank you very much.